Thank you for coming to the workshop today. We're going to be doing how to enliven your online biology instruction. It's co-presented by 80 Instruments, and we're joined by Liam and Charlotte from their business and John Melville with Vernier. We're excited to tell you more about the 80 Instruments LT biology collection, which you'll learn about today, and show you how this platform integrates with Vernier sensors and data collection. We will be using the Q&A feature today for answering questions. So we do want this to be interactive. So we encourage you to put your questions in the Q&A. And we'll also have this recording available on both of our company's websites after the webinar today. So you'll be able to access that. We'll put a link in the chat feature later on to show you where you can find that. So I wanted to get this interactive. So we'd like to launch a poll to begin with. And so if you don't mind taking a few moments to answer the two questions that we have, what we'd like to know are you familiar with 80 Instruments? And then what level do you teach? Would be helpful in determining what we talk about in the presentation today. Looks like we're getting some results here. I'm going to go ahead and end it, but it looks like the majority of our participants today are familiar with 80 Instruments. And it looks like that we have a good number of attendees who are four-year college professors, and then also some two-year and other. I'm going to share my screen now. Okay, so wanted to go over the agenda real quick. So we just did the quick poll, and I'm going to introduce the presenters again. So John Melville from Vernier is going to be showing you the platform and explaining how this integrates with Vernier products. And Liam and Charlotte from 80 Instruments will be on standby to also answer questions. They'll be giving you an introduction of the LT platform and specifically the biology collection. And we will have time at the end of the session today for questions and answers. So we will be answering those. And if we happen to not get to your question during the session, we'll follow up with you afterwards. So with that, I would like to introduce Liam.
Hi everyone, I'll just start sharing my screen. All right, so yes, as Angie mentioned, I'm the Customer Success Manager for 80 Instruments, and I specifically look after institutes using LT in Australia and New Zealand. And so I'm just going to give a quick overview of what LT is before handing over to John and Charlotte to delve more deeply into the biology collection that we've been working on between 80 Instruments and Vernier. So just taking a step back, and trying to bring LT into the context that you may be familiar with, or your environments that you're using currently, which I assume for many of you is your learning management system, maybe Moodle, Blackboard, or Canvas. Is pointing out that LT is not one of those, but it's designed specifically for authoring content and having a really nice, clean learning environment. And what it is, it's an online learning platform. So essentially, you just go to a website, both students and educators log in the same area. And I'm on the login page here purely to highlight that there's two ways to log into LT. And that's single login and group login. And the reason why I'm mentioning that is that it's just showcasing the versatility of LT, that it can be used both for remote teaching, but also for in-class activities. And given that the biology collection has been designed with labs in mind, the group login feature is going to come into handy there. So what it is that students can all log in on a single device, record whatever they're doing in that particular lab, and then they can leave that lab and each individually access the data that was recorded as a group, and then they can be answering questions by themselves. So it's really starting to blur the lines between in-class and out-of-class labs, which is pretty cool. So I'm not going to do the awkward try and type in and forget my password. So I'm going to go into an environment where I've really logged in, and you can start to get an idea of what LT looks like from a student's perspective. And a couple of things to point out is that on the left here, we've got the list of courses that are available. And this is a showcase, what's called instance of LT. And so normally students won't be seeing such a long list of courses. It's just being utilized to showcase the different collections that ADI has developed. And the one that we're focusing on today is the latest biology collection. So once you've selected the course, you can see all the content that is available to the students. And ADI, we're pretty pedantic with having these lovely little thumbnails. I love this one here, kind of looks like a little Elvis gut. So I'm just going to delve into what's called a module here. So you'll see that all these icons have 2 lessons or 4 lessons. That's just indicating that it's a group of lessons. And actually I might jump into this one just to show that, yeah, you've got these 4 lessons that are all themed around the central nervous system. And so when the student wants to come in and learn around central nervous system, for example, they can go into any of the lessons that they want. So just to give you a quick intro. And so once delve into the lesson environment itself, you get an idea of what it looks like. So it's very clean environment, and it's laid out in a linear fashion where students progress through a series of pages that are theoretically infinitely scrollable. Obviously, it's not good pedagogical design to have very long scrollable pages. So obviously I want to keep them relatively short. And on the pages, we have interactive elements such as pop-ups. So you can start to have these little branching elements to a page, which is quite useful. And then students can be navigating through lessons and seeing images, interacting with many pages here, and large elements or common elements to LT questions as well. So scroll through and you can see that we've got example of question here, table, and so you can be typing in whatever you want. And there's also drag and drop features, which is a common theme to LT. So you can be dragging and labeling. Horribly labeling this one, definitely not correct, but students do have the option to check the answers and then realize that maybe they should do a bit more study. And so forth. So just giving you a brief teaser, so say of what's capable in LT and what students are seeing. And before I hand over to John, I'm going to go into another area of LT, and this is the staff side or the educator's side of LT. So kind of going behind the scenes so you can get an idea of how easy it is to import content from the collections that ADI have made available, including the biology collection that we'll hear about later. It's as simple as clicking on the collection that you're interested in, scrolling through the content and going, oh, that looks interesting, and then just importing that into your particular area of LT. And if you want to be making any edits to the lessons, they're all fully customizable. We don't want to be restricting how you want to deliver the content. We just want to do a lot of the heavy lifting, get you the content that you want that you can use out of the box, or if you want to do slight tweaks or even create full lessons from scratch, you can do that. So if I was to, say, open up one of our starter lessons for students, then you can see just how easy it is to be making edits to lessons. So as an author or an educator, you'll come in and you'll see their environments pretty much as a student will see it. So it's very nice to know that what you're seeing is what the student's going to see, so you're not having to kind of imagine what it's going to be like. So any change you make is what the students will see. And once you click edit, you'll see the authoring tools appear. And we want to make it as simple and intuitive as possible. So our development team decided that drag and drop was the best way to go about this. And you may have seen it in other tools where you can click on different elements, or in terms of LT, what we call panels on the page, and you can actually shift them around, and everything dynamically resizes to cater for whatever changes you've made. So you don't have to worry about, hitting enter, that many times, or, spacebar just to get that perfect alignment. LT makes it far easier. You can just drag it around, and you can be double clicking in to edit the content of that particular panel. And you can also add new panels from the panel library here. So it's a range of basic panels. You've got questions, so at the moment 9 different question types, video and audio. And importantly for lab purposes, there's the sampling option as well. And it's as simple as dragging one of those on and typing it in. You can also drag on elements from outside of LT into LT directly. So say, for example, I wanted to drag on the image again. You can quickly drag it from outside of LT into LT, even choosing what panel type you want as well. So that means it's very easy to be transferring content from where your resources lie currently, whether that's maybe a PowerPoint, PDF, Word doc. You can just quickly transfer it into LT, and away you go. So I don't want to be taking up too much of the time, but I'm happy to answer any questions, maybe towards the end of the webinar. And if, don't get around to answering questions during the webinar, happy to try and answer them at a later date, or you can visit our website and explore it for yourself. So with that, I'll probably look to wrap it up there and hand over to John to delve more into the biology collection. Cheers.
Cheers. So I'm John Melville. I'm a former college professor. I got my BA in biology and then psychology at Sonoma State University and a PhD in zoology. I did postdoctoral research as well, but most importantly, I'm a former educator, like probably a lot of people here. And I've taught majors neuroscience, general biology, marine biology. I've taught a lot of courses. And my main focus, though, was on active learning with an emphasis on data collection and then also inquiry. And back at Oregon State University, back in the 90s, I was one of the first people to introduce data collection using 80 Instruments equipment, but I also really loved Vernier and their data acquisition system as well. So I got really excited when it looked like we were going to be able to work together to produce this biology collection. And the biology collection is terrific. It consists of 19 labs, and these 19 labs are developed from some of the Vernier books, but they've been expanded and modified, and Charlotte, her team, has done a great job. Has done a great job with all of them. I'm going to increase my volume there a little bit, so hopefully you can hear me. The biology collection integrates with Vernier GoDirect sensors, so it's one of the only platforms that exists where you can actually look at remote data, and you can also collect in real time in the platform. So it's suitable for both remote learning environments, and we have a great deal of example data built into it for you to analyze as well. And then one of the key things that I'm going to do is I'm going to take you through the Enzyme Action lab, but that's after I take you through showing you some of the things that are on or about the biology collection. So with that, I'm going to share my screen. Sorry, that's going to stop sharing other screens. There we go. And right now I'm on the AD Instruments webpage. And I just wanted to show you here. I'm on the LT webpage actually. And what you can see is they have all of these great courses: human physiology, clinical medicine, nursing, animal physiology. And then down here we have biology. And I'm just going to click on that to see inside. So this is the LT online learning platform. There's this great little video that gives you an introduction to the 19 experiments, how they all work, some great features on how to use LT, the biology content, customer stories, and you can even do a preview or get a free trial very easily here. I just want to go through a little bit of the content and show that to you. So I'm going to scroll down, because if you take a look at, let's just say, we're going to do enzyme action. If we take a look at cellular respiration, there's a nice intro to that lab activity, gives you an intro. But down below, it also tells you what you would need to do that experiment. So right here, you would use the GoDirect CO2 gas sensor. But AD Instruments also sells a very specific package. So they have the GoDirect biology sensor package, which includes all of the instruments, all of the GoDirect sensors that you would need to do the entire collection. So you can either figure out which ones you would want to purchase to do some, or you could buy all of them so that you could do the entire collection. All right, so what I was going to do next is just show you what LT looks like on my end. So I'm here in my course. So I have imported an entire biology course. So I've labeled it, here's John Melville, I've labeled it Bio 1. And I've imported all of the courses from the biology content. So you can see there's acid rain, animal behavior, aquatic photosynthesis. So all 19 labs I've imported in here. And there's a student view. I'm in the administrator view, but you can also invite students to use this. So I'm going to use this the way a student would see it. So I'm going to move to student. And here's the recent lesson that I've published that the student could use. So we're going to go through this as a student. So if I tap on here, I'm just going to say start this lesson. I'm going to start it by myself. And what you can see is that they have this great introductory information in addition to just this enzyme activity. And this enzyme activity testing catalase is a standard lab that many of you have probably done, which looks at enzyme action using catalase and hydrogen peroxide. But AD Instruments has done a great job with giving us all of this information up front, talking about what enzymes do and hyperlinks that you can click on. Even really nice and beautiful figures, shows the reaction, etc. So there's a lot of what I'd like to call rich content that exists inside the platform. In addition, the learning objectives are laid out really nice. So you can see these are the learning objectives. We're going to use the GoDirect oxygen gas sensor to collect data. We're going to look at increasing enzyme concentration. Then we're going to look at increasing substrate concentration. And then also there's a Lineweaver-Burke plot that they can do. Now I'm just going to do the increasing enzyme concentration and show you how to analyze data, because it would take a very long time to do this entire lab since it's designed for a full lab period. What's also really nice is most of these activities have a challenge. So it starts with a challenge. So they're going to ask me to label this diagram. So I could do things like label the active site. Where's the enzyme in this graph? Okay. Where's the enzyme product complex? Well, that might be over here. Where are the products? And this is really nice because it allows the students to go through a challenge so you can test their knowledge before you get into data collection. So you can make sure that they are understanding the key concepts before you move into the other parts of the lab activity. And a lot of other lab platforms, we just go straight into data collection. You hope you get data, and then you talk about it all later. And what's really nice too is you can check the answer here. And you can see I got 100%. That's really nice. That's probably because I've been practicing for a while. There's also things down below where I can look at which one of these is true. Do enzymes affect the amount of activation energy? Do they increase it? Do they reduce it? I'm just going to say they don't. It says that's incorrect. So that, you know, even if you select a wrong answer, it will also give you the information that your students need. And in addition, we can formulate a prediction. So based on our knowledge about enzymes, we can, you know, I can write in a prediction on what I think is going to happen. So there's a lot of information and material here that makes this a great platform for teaching. So here's the material setup. I have, it shows us how we're going to set the system up with an oxygen gas sensor, a stir station, and an Nalgene bottle. And then we connect the oxygen gas sensor to get started. And just so you can see, I have that right here. So hopefully you can all see that. I've got an oxygen gas sensor right here. I'm going to begin collecting. I hope that's not too noisy, but I'm just going to have it spin there, or I'll have it spin there in a minute. What I have over here is I've already put in 5 mils of water right here, and I'm going to add yeast is going to be my enzyme suspension. Another thing that you could use, which is really useful, is this is purified catalase from Flynn Scientific. Purified catalase is great because you don't have to worry about proofing the yeast or anything. And you can make a little concentration of that yeast suspension that'll last a long time. Charlotte will wind up talking to you about how the instructor information is also located in LT in a great place. So I don't want to steal her thunder. So we're basically, next one I'm going to do is I'm going to do part of this experiment. So we're going to go to the next page. And right here it says, you know, set it up, label some test tubes 1, 2, 3. I can wear some safety goggles, you know. I should put on some gloves. I'm going to put on 1 glove. And actually what I'm going to do is I'm going to turn off the video so you don't see me moving back and forth here. But you will see the video over here, and then I'll come back. So just give me a moment to do that. I'm going to scroll down because the reaction is going to take place right here. We're going to see the experiment there. There we go. It's really nice how it just resizes. Thank you for your patience as I do this here. Great. So I'm going to stop my video. I promise I'll be right back. So what I'm going to do now is I have the little stir station running. I have my substrate, which is just 3% hydrogen peroxide. I'm going to add that to the 5 mils of water after I add the enzyme. To make sure that the enzyme reaction happens rather quickly, what I'm going to do is I'm going to add 4 drops so that we see a nice response, not just 1 drop. And I've got this spinning here. So I will pick up my yeast. And I will add 1, 2, 3, 4 drops. Now I'm going to add the hydrogen peroxide, and I'm going to invert this test tube. Then I'm going to pop the water, the solution, in here, and then put the oxygen gas sensor on top. There we go. Invert it a few times. Put this in place. And then I'm just going to wait for 30 seconds. Turn my video back on now, so we'll do that. The reason why we wait for 30 seconds is to allow the oxygen gas sensor to re-equilibrate. There we go. There we go. So that's at 20%. And we let this run for about 2 minutes and watch it slowly increase. Oh, there we go. Increasing a little bit. Stretch that out there. And if I wanted to make this more exciting, I'm going to just add a little bit of my purified yeast suspension just for fun. There we go. And this reaction will last for, normally do this first reaction for around, it's 200 seconds, so 3 minutes and 20 seconds. If you're ever doing this experiment, it's really important to proof the yeast or to proof your enzyme to make sure that you get a nice response. There we go. Now we're beginning to go up. Very nice. Perfect. Well, I don't want to have you all sit here and like watch paint dry or watch an enzyme react. So I'm just going to hit stop. Normally we would let this run for, I'll let it run a little bit more so you can see that it's working pretty well there. Oh, I could add a comment, right? That's really important here. I could say, oh, that's 4 drops. There we go. That's beautiful. So now I'm getting a really nice increase in enzyme reaction there. Wonderful. Very nice. What's great is down here, you can see how long we've been recording right down here in this bottom corner. It's almost 200 seconds. I'll let it go a little bit longer. Great. So I'm going to hit stop. Now I will turn this off so that you don't have to hear that irritating rotating sound. Now, what's great is that on the next page, if I go to the next page, we could analyze this. So they've got a really beautiful way, and let me open this up so we can see this a lot nicer. There's a beautiful way to analyze this data by dropping a marker and then looking at the change. But all of you are, probably a lot of you are also teaching remote. And one of the beautiful things that 80 Instruments has done is they've taken sample data that we provided them, and it actually lives in the LT platform. So if I hit Shift and Start and select example data, it's going to ask for an access code that I know. And then it's going to come through with some beautiful sample data. And I can increase this here, scroll across, let's increase that a little bit more. There. And now what I just want to do is show you how we can actually analyze this graph. AD Instruments has done a beautiful job at creating a table, analyzing the data in a really simple way, but a very effective way. So I'm just going to drop this marker where we first added some drops. I am then going to insert this little marker, and I can drag this marker to the end of the data collection. And you can see that the change was 1.28% over this time. And if I just drag these into these columns, you can see what's beautiful is it automatically calculates the reaction rate. And then I can go to the next run, and I can do the same thing. I can take the marker, drop it, take the marker and drop it. There we go. And then I'm just going to grab the tab over here. And you can see for this new run, it's 2.83% for 173 seconds. So I can just drop that into the table. So these tables are truly interactive. Unlike having students go out into the lab manual and write everything down and then come back, these tables are truly interactive with the graph, which is something that I just think is really amazing. And then I'll do the last one. I can drop a marker. And then I can put this here. And then down below, what you can see is it's actually also created a graph. So it's automatically created a graph. I could title the graph. I could say reaction rate, concentration. I can even edit the graph. And if I wanted to, I could put in linear fit. And then there's even questions to test the understanding here as well. So it's a really, really rich platform that integrates data collection and/or sample data. And let's see, right, test our knowledge, examine your data closely to make a conclusion in the box below. There's a question about the independent variable, observe your results, etc. Then the next thing that happens in the lab is you can take a look at the effective substrate concentration, which is something that a lot of people want to look at, which is, well, how does substrate concentration work? And if you've ever looked at or done this, the big things you want to look at are KM, Vmax, VM, things like that. So you can actually do that as well. There's a way to get the sample data in as well. Same thing, if I were just to hit Shift Start, sample data would come across. And once again, there is a whole other aspect of this lab that looks at, well, here's the actual data that you could analyze. And once again, it would also plot the data so you could do a plot. And then there's even a really nice introduction to enzyme kinetics, which uses that data that you've collected, where you can actually calculate Vmax, half Vmax, Km, and look at all of that using a line mover BERT plot. So, and this automatically uses the data that's in LT. So you can either, once again, collect your own data, use the sample data. And one of the beautiful things about LT is that if this is too complicated for your class, you can remove that. If you just wanted to look at one part, you could edit that and change it. And once again, there's going to be a test of understanding here as well. There's a review and integration. So once again, there's a really nice section that reviews student knowledge. Do you understand how everything worked? What questions do you have? How did everything increase? So it's a very thorough and rich platform. But even beyond that, there are even things that go beyond just the classic, what I would say, data collection, sampling, and analysis. There's even extensions that look at experimental design. So if you wanted to do inquiry or you wanted to add other components, the content developers that have worked on this have put a tremendous amount of effort into it so it can be even richer. And with that, I would like to hand this off to Charlotte that can talk about some of the curriculum design and all of these really amazing features that have also been built into LT with this in mind.
Hi, everybody. Thanks, John. That was great. My name's Charlotte. I'm an instructional designer on the LT content team at AD Instruments. So an instructional designer creates educational materials and experiences using pedagogical frameworks. I recently managed the development and the release of the LT biology collection. And when this collection was proposed, the team was really excited because we were thinking about what do we know about the biology undergraduate curriculum, what can we do with this collection to make it appropriate for the market. And we worked on it between around October 2019 and it was released in August 2020. What makes the collection really exciting is that we've taken Vernier's lab manuals and their sensors, their Go Direct sensors, and combined them with the LT platform and with our content design skills. And I think what we've created is a really dynamic and diverse collection of lab activities. So I just wanted to talk to you briefly about the highlights of the collection and the development process. So when we began working on the collection, we pretty rapidly identified this report by the American Association for the Advancement of Science. So in 2011, they released a report called Vision and Change, a call to action. And this essentially assessed the state of biology undergraduate education and found that the curriculum could be very heavily fact-based and that it was lacking in opportunities for students to be exposed to real-world science. So they thought that what would be important would be focusing on broader concepts, for example, evolution, and then building facts underneath that so that students had a better grasp of the wider landscape of biology as a field, and also ensuring that students become capable biologists by exposing them to real-world research opportunities. So they identified 5 key concepts and 6 core competencies. So out of all the insights that were available in that report, we thought that the 6 core competencies offered a really workable way for us to integrate their findings into the collection. So I'm not sure if any of you are familiar with the report, but the 6 competencies were the ability to apply the process of science, to use quantitative reasoning, to use modelling and simulation, to tap into the interdisciplinary nature of biology. So knowing that biology is informed by all these other realms of knowledge, as well as its applications in the real world. Communicating and collaborating with others. So having students know that they might be communicating with industry or with the government or with the private sector. And then the ability to understand the relationship between science and society. Another way that we decided to sort of scaffold this collection, we really went back to the basics. So I'm sure many of you are familiar with Bloom's taxonomy. So Benjamin Bloom came up with Bloom's taxonomy in the mid-20th century. And initially it was a series of scaffolded cognitive skills that could be applied to assess learning outcomes. And increasingly, recently, it's been used to classify assessment questions as well. And that's what we have done in the biology collection. So Bloom's taxonomy is usually viewed in the shape of a triangle. So you've got a broad base and you're building on that and getting narrower as you go up. So the basic skill is remember, and then you move to understand. In the middle, you have apply, and then you're moving through analyze, evaluate, and create or synthesize. So Bloom's taxonomy has been revised, and there can be some argument between where evaluate and synthesize sit. But what we wanted to do was to use that scaffolded tool. It's widely agreed that these skills build on each other to scaffold the collection itself. So I just want to, and before I move on to actually showing you some lessons, there was a very interesting study that actually looked at assessments provided by 50 educators who teach undergraduate biology, and they found that 93% of the assessment items focused on remember and understand, which are those 2 most basic levels of Bloom's taxonomy. So when we saw that, we thought, wow, what can we do with the collection to make it a little bit more rigorous and have students applying and analysing and synthesising. So I think I'll just share my screen now. So during the development of the collection, so John's just shown you Enzyme Action, so I have aquatic photosynthesis here. We use Bloom's taxonomy so that when we created an assessment question, we always ranked it with a Bloom's taxonomy score. And we also implemented another tool. So we looked at scientific writing. So scientific writing requires the writer to provide data to support their arguments. They can't just rely on an opinion. And scientific arguments are strongest when they're linked to scientific principles and evidence. So there's this framework that some of you might be familiar with called claim evidence reasoning. So this is a tool that's in the past been used in middle school, so sort of grades 5 to 8, but increasingly it's sort of making its way into the tertiary sector. And I have an example of claim evidence reasoning in this lab. So as you move through the lab, generally on challenge pages that John showed you before, we would look at basic levels of Bloom's, so remember and understand. When you're looking at activity and analysis, you're looking at applying their knowledge and analysing the data. And then on review and integration pages, we aim to have some higher level questions like evaluate and create. But if we look at the analysis page here, at the bottom of this activity page, there is a claim evidence reasoning activity. So this lab looks at photosynthesis and cellular respiration in aquatic plants. So first we emphasize the importance of a scientific argument. So students, if they're struggling to understand why they have to present their information like this, we're justifying why we're asking them to do this. And then we ask them to make a claim. So based on their data, they have to make a statement on whether or not aquatic moss performs photosynthesis and or cellular respiration in dark or light conditions. They have to talk about the evidence, what's led them to that claim, and they should be able to do that with their data that they've pulled out of their panels. And then reasoning. So through reasoning, we want them to link their claim to establish scientific principles. And then finally, some frameworks of claim evidence reasoning don't actually include rebuttal, but we thought it was a really useful skill, and it also links to higher levels of Bloom's taxonomy like evaluate. So if somebody comes to you and says, how can you back up your data, that students are able to rebut. So we've said here, are you confident that it was the aquatic moss, and how are you sure that it's not just an environmental artifact, and asking them how they would reply. So it's really important to emphasize to students that you don't just do an experiment once and it's perfect and you can trust it, making sure that they know that there's experimental error, there's machine error, things like that. And then we moved on to general structure. So John actually looked through the lab with you earlier on, so you already know about the general structure of the labs. We have that introduction. Introductions, generally, we want to provide a hook for students. So we want to increase student motivation, so they want to move through the lab. So we try and introduce either a question or a real-world application of knowledge or the general concept of the lab at the start. On the challenge page, like John said, we have 2 to 3 questions that look at basic information, so asking them to recall their knowledge, what they might already know about the topic. And then the activity and analysis pages are really the meat of the lab. And then finally, on that review and integration page, we ask them to revisit their prediction and make a conclusion. So it's really important for students to know that perhaps what they thought wasn't correct and that they're able to revise their mental models. And then these questions, like I said before, we talk about higher levels of Bloom's taxonomy, so aiming for analyze, evaluate, and create. And then finally, we ask students to reflect. So like I said before, we want to emphasize to students that it might not always be perfect. So results of scientific experiments generate new ideas and questions. So asking what questions you have or ideas for additional variables. And actually that brings me to guided inquiry. So in the diffusion through membranes lab, we introduced an extension. So the basic activity in this lab is reasonably simple. But we wanted to give students, as outlined by the Vision and Change report, an opportunity to actually participate in scientific inquiry. And we do realise that that can be quite hard in a practical setting because we're not sure perhaps what materials you have available to you in the lab. With certain students, you don't want them having free rein because they might go totally off the rails. So we wanted to provide bumper bars, if you will. So we have guided inquiry. It's not quite free range, but it lets students pick a question. So they would pick one of these questions relating to the central topic of the lab, and then they would look at parameters of the experiment. So we really want to introduce them to the scientific method, and we get them to identify each of these parameters for their chosen experiment. and then we want them to make a prediction and explain why they've predicted that. And then they have to come up with a method to investigate that question. And then we have a page where they collect their data. So the data sampling panel on this page is set up so that when the sensor is connected, the sensor that they used for this lab, they'll be able to collect their own data. And then they're moving through, there'll be a table that they can set up themselves, and then they have to be able to set up their own graph, and they can edit the graph. So before John was showing you an auto-populated graph. With this edit graph option, students can choose what type of graph is most appropriate for their data, and they can also apply curve fits. And then they need to make a conclusion and explain why their results support their conclusion or if they don't. And then finally, we generally have a little test your understanding section in these guided inquiry activities. And we quite like often to present questions that maybe say, here's a data set, explain what's wrong with this data set, especially if students are working from home or they're not working in large groups. This allows them in some way to participate in peer review. And then finally, we have this lab report. So initially, when we were developing the collection, the lab report page was just for the extensions, but we realized that it's so useful for students that we've incorporated it into every lab. But this essentially guides students through the scientific method. So we're talking about, they have to talk about the title and authors, they have to write an introduction, methods, results, conclusion, and then provide references and appendix. So it's guiding and supporting students through this process, but this is a really stereotyped way of scientific communication, and it's really important to introduce students to this if possible. So we've found that this page has been really useful. And of course, we know that students might come from different backgrounds, or your lab periods might be of varying time periods. So these extensions are removable. So if you come in and you think, no, I don't want people to be investigating their own question, you can remove that from the lab. And then finally, we provide a supplementary resource for educators. So this is called an instructor's material. You can access it through the LT instance, and there is one that's written for every lab. So first you'll have a little introduction, a synopsis of the lab, if you will, and then we walk through the equipment list and alternatives. So this is what students see in the lab as well. And then we have notes to educators. So these are really handy tips that have come from Vernier, and some might have been added by us, but talking about how you can best run the lab and make it go as smoothly as possible. And then finally, and also we include references. So for some of the techniques we've used, claim evidence reasoning, we talk about where that's come from if you want to do some further research. And then finally, you will have seen the raw data, sample data in those panels in LT, but we also have example data in the instructor's materials. So where students can't get to them essentially, but it's giving you this data so you'll know what goes in the tables and what example graphs might look like. And example data was a key facet of the biology release. So we pre-released biology during the middle of the COVID-19 pandemic, and we realized that example data was really important to people who might not be able to get onto campus, but for educators who still want students to have some experience of data analysis and to know what it's like to be in a lab completing an experiment. So we're really proud of our suite of example data that's sitting in these lessons. And yeah, I think that's all from me today, but we're excited about the biology collection. We want students to be engaged with biology, and we want educators to have a streamlined experience. So thank you for taking the time to learn about it with us today. And I'll pass back to Angie for questions.
Hi, everyone. So if you do have questions, we don't have any currently. I'm going to ask our panelists, are there common questions that come up about the platform or integrating Vernier sensors?
I would say I can answer integrating Vernier sensors. The key thing to know is that you need to use they only work with the GoDirect sensors. So the sensors do have to be the GoDirect ones. It also only works right now, I believe, Liam or Charlotte, you can tell me, over USB and on PC. But the sample data exists across all instances. So the sample data is there across everything, even if you're on a Mac. But to actually collect data into LT, you need to be using the USB port on the GoDirect sensors, and you need to be using the PC platform. Now I think Liam or Charlotte might know more about the classic questions that people get with how to use LT and authoring and things like that.
Yeah, I could probably jump in a bit on that one. I should point out that it's very easy to set up a trial if you are wanting to explore LT. So that's probably the thing I'd recommend the most. If you do have questions about LT and functionality, is that it's very easy to get access to a free trial, so you can have a go with that. And we can even do that with students as well. So some common questions. Generally, it's kind of how LT is going to be integrating into your current system. So just pointing out that it is very easy to be linking out from your learning management system, or wherever you currently have students engaging with content, that you can simply insert a link to LT and students can, link to LT in your LMS, and students can be navigating out into that environment. And yeah, there's a lot of different avenues that could go down, but I'm just checking there's no questions that popped up at the moment. I'm more than happy to talk offline if people want, if they're too scared to be asking questions on a live webinar. Not sure, Charlotte, if you had any maybe around the content in particular.
I think sampling is generally an area where we'd have the most inquiries, and I think John has covered that quite well. So yes, live sampling would be over USB connection and Windows PC, but that example data that you saw is available no matter what, because it's in the cloud. It's just available via the browser.